Living positively, living positive, living positively, living positive.
Welcome to Period Treatment Literacy, the comprehensive video series on all aspects of HIV, its treatment and care. My name is Lihle Dlamini. Treatment literacy is not only important but vital because experience shows that treatment works best when people with HIV, lay counselors, and healthcare workers are all equally empowered with knowledge about HIV, so that all can work together in beating the challenges of the HIV/AIDS epidemic. With knowledge of treatment, we can identify illnesses early and make sure they are effectively treated by our healthcare workers. Treatment literate people with HIV are happier and more confident. Lay counselors and healthcare workers are less stressed and able to work better. What does HIV mean? HIV stands for Human Immunodeficiency Virus. So, HIV is the virus that kills the immune cells that defend our bodies from infection.
What does HIV mean? HIV is the virus that causes AIDS. H stands for human, I stands for immunodeficiency, and V stands for virus.
It is very difficult to be sure exactly where HIV came from. There are different theories. The best theory is the one for which there is most evidence. Scientists have discovered that HIV appears to be a combination of two other viruses, which are found in the West African monkeys called the Sudi-Mangabe. These monkeys are hunted and eaten by chimpanzees. At some point, anytime up to 1 million years ago, these two viruses met and combined in the body of a chimpanzee, creating a virus called SIV, or Simian Immunodeficiency Virus, which is very similar to HIV, the Human Immunodeficiency Virus. At some time in the last 50 to 80 years in West Africa, the simian or monkey immunodeficiency virus crossed from chimpanzees to humans. This crossing of diseases from one species to another is not unusual. Bubonic plague, for example, which is described in the Bible, is mostly a disease of rats, but it infects humans when we are bitten by fleas from those rats. Rats carrying bubonic plague are found in America, Africa, and Asia, and used to be found in Europe. When it first arrived in Europe in the 14th century, it killed 1/3 of Europe's population.
The origin of HIV. It is common for germs to cross from one species to another, creating a new germ in the process. Humans were infected with SIV from chimpanzees, creating HIV. New infections that humans have not been exposed to before often cause people to be very sick or die.
did HIV come to spread around the world? Analysis of HIV seems to show that the first human infections occurred in 1930 in the Democratic Republic of Congo. Over the next few decades, the infection spread amongst small populations in Africa. By looking at old samples of blood, scientists have confirmed that a man in DRC was infected in 1959 and that the first HIV infections in the United States happened in the late 1970s. By 1980, HIV had spread to every continent besides Asia, but it was only identified as a distinct disease, AIDS, in 1981. At that time, no one knew what caused AIDS. The human immunodeficiency virus, HIV itself, was only discovered in 1983 by laboratories in the US and France. But the epidemic moved faster from the late 1980s onwards. The movement of people along trucking routes, the movement of refugees, and through business pushed the epidemic south from Central Africa. Now the epidemic moves in all directions. In Lesotho, Botswana, and Swaziland, over 30% of the population are infected. According to the UN, in sub-Saharan Africa as a whole, over 26 million people are infected. The prevalence among adults is 7.4%.
The history of HIV. An epidemic is a disease that affects many people in an area. For example, in a country like South Africa. A pandemic is a disease that affects people in many countries. Prevalence is the number of people living with a disease compared to the total population. Sub-Saharan Africa refers to all African countries south of the Sahara Desert. The first AIDS death was thought to occur in 1959. AIDS was recognized as a new human syndrome in 1981. A syndrome is a collection of disease. HIV was identified as the cause of AIDS in 1983. In 2006, in sub-Saharan Africa, 24.5 million children and adults are living with HIV.
Overall, at least 11% of all South Africans are infected. Some surveys claim the number is as high as 14%. In the age group 30 to 34, the infection rate is 24%. In 2005, more than 5 million South Africans were HIV positive. This is twice the number of people that live in Cape Town. In 2005, more than 320,000 died of AIDS. More than 800,000 people need ARVs now. The government says they are giving free ARVs to 175,000 people, less than 1 in 5. However, even this may not be true. We know this because the company that sells ARVs to the government records how many tablets it sells.
Number of people living with HIV in South Africa. As many as 5.5 million of South Africans are living with HIV. More than 700,000 people are living with AIDS and need ARVs. Over 130,000 people are receiving treatment from public hospitals and clinics.
Throughout this series, we will be dropping in on the Beat It Treatment Literacy Workshop, where today's facilitator, Vuyiseka Dubula, is discussing each aspect of HIV. Let's meet Vuyiseka and the workshop group as they talk about the different kinds of HIV.
My name is Vuiseka Dubula. I'm going to do an introduction to HIV. HIV belongs to retrovirus families, and retroviruses, it's a family of viruses that can multiply themselves. But what I want us to discuss with the retroviruses and HIV is the fact that we have about two types of retroviruses. One is HIV-1 and HIV-2. Which HIV do you think in South Africa it's the most common HIV?
I think here in South Africa we've got HIV-1, and HIV-1 is more aggressive.
Usus Nomozuko is telling exactly what I was going to tell you, that the HIV that we have in South Africa, it's HIV-1, and it's the more aggressive one. That is the one that is responsible for a large number of people who are living with HIV in South Africa and some parts of Africa. But HIV-2 is not common in South Africa. We found HIV-2 in other parts of Africa.
HIV-1 is the most common. HIV-1 is also divided into groups called subtypes. The most common subtypes are B and C. Subtype B is most common in North America, Europe, and Australia. Subtype C is most common in Africa and India. It is subtype C that has caused the worst epidemics and is responsible for half the world's infections. Here in Southern Africa, most people are infected with subtype C, but there are still many differences between HIV in different people's bodies. These different kinds of HIV are called different strains. It's because there are different strains that it's so important to avoid being infected more than once or being reinfected. Back to the workshop.
But that does not mean if you have HIV-1, you cannot get HIV-2 at the same time. If you are having HIV-1 and you sleep without protection, protection means a condom, you can get reinfected with another HIV, maybe HIV-2. So that means you will have two viruses, and then the one that is more aggressive would want to multiply more than this one. So you have two viruses, but how can you control two viruses? So it's going to be difficult to control two viruses in one body. That is why you have to use protection or to use condoms so that you don't get reinfected with another HIV.
There are two types of HIV, HIV-1 and HIV-2. HIV-1 is the most dangerous, and it is the type that is most prevalent in South Africa. HIV-1 is divided into different groups called subtypes, A, B, and C. There are different viruses or strains in each subtype. If you are HIV positive, you should still use a condom when you have sex. This is because you may still contract a different strain of HIV.
Is there anyone in the audience who can tell me also how can you get HIV? Nikki?
Okay, HIV can be transmitted in two ways, like through our blood and through our body fluids, like vaginal fluids and semen from the males.
Thanks for the answer. That is true. You can get HIV mainly through blood and body fluids. Body fluids being semen from the males and vaginal fluids from the females. How can you get HIV through blood? What can be the things that you do that can put you at risk of getting HIV through blood? By using the same needle. If you use your bit of the drug and then you want to share it with another person using the same needle, you can get HIV through that because the HIV will stay in the top or in the tip of the needle. Then that other person can get HIV. That is if you have HIV or if the other person has HIV.
I think a doctor can be at risk when maybe he's operating on an HIV positive patient and he cuts himself with one of his tools and he gets into contact with the patient's blood, thing like that.
That is also true. A healthcare worker is at risk of getting HIV through blood. For example, Lucy has made an example that a doctor is busy in the theater and some of the tools that he's using or she's using can maybe, if it's a needle, you can get a prick. If it's a scissor, you can get a cut. With the blood that is contaminated with HIV, you can get HIV. Say, for example, the patient needs some blood transfusion. If one of the blood is not tested, you don't test the blood, you transfer the blood to the patient. Sometimes the patient can get infected because you don't know whether the blood is contaminated with HIV.
HIV-infected blood enters the body through cuts in the skin. HIV enters the blood circulation. Inside the blood vessels, we find red blood cells and white blood cells. HIV can float freely in the blood or rides along attached to the white blood cells.
The other way of transmitting HIV through blood is when the HIV positive woman is delivering the baby. There is more blood involved. Maybe the child might get the cuts from when the time the mother is delivering.
Sis Ntombi said it all, that also an infant or a small baby can get infected through their mothers when they are delivering, because there might be some cuts happening when the baby is being delivered or being pushed out, so the baby can get infected with HIV as well.
An HIV-positive mother has HIV in her breast milk. Normally, this is fine as the HIV will just pass through the child. But the lining of the baby's stomach and guts are very, very sensitive. Anything other than breast milk, even water or medicine, will irritate the guts and make it possible for the baby to absorb the HIV in the mother's milk.
You can get HIV from vaginal or anal sex, mother-to-child transmission during birth or breastfeeding, through blood from needle stick injuries, sharing needles, oral sex, which is a low risk. You can't get HIV from kissing, touching, sharing dishes, mosquito bites, coughing or sneezing.
Now, can we move on to the other body fluids, the fluids that you told me about, vaginal fluids and semen? Who are at risk there? Who can get infected? Okay, Sisuki.
There, I would say everyone who have unprotected sex is at risk of getting HIV. It's either a woman, it's either a male, it's either a child, a youngster, a prostitute, or if someone is having sex without using a condom.
Usisuki has summarized all the risk groups, that it can be a gay man, it can be a lesbian, it can be a straight person who is having, a woman who's having sex with a man, or a man who's having sex with a woman can be at risk of getting HIV because of the vaginal fluids and the semen. It can be sex workers, it can be truck drivers, anyone who is having unprotected sex or unprotected intercourse can have HIV.
Who is at risk of HIV infection? All people who have unprotected sex. Women are more at risk than men. Children of HIV-positive mothers can contract the virus from their mothers. Sex workers, gay men, lesbians, and men who have sex with men. Mobile populations such as migrant workers and refugees. Injecting drug users.
body is built from millions and millions of tiny cells. Different cells have different functions. So, the cells in our skin are different from red blood cells, and brain cells, and liver cells, and CD4 cells.
Our bodies are made up of millions of cells. Because we have so many cells in our bodies, and those cells are helping us in fighting infections, they are helping us in transporting blood, in oxygen, so we need those cells. But to imagine the millions that I'm talking about is to imagine a sea. A sea is made up of water and the sand. If you take the sand and you take one particle of the sand, that would represent one cell in your body. So you check all the million particles of the sand in the sea, that means you have so many. We can't count them, but they are there and we cannot survive without them.
Our bodies get attacked by germs or pathogens. These are things which cause illness. Bacteria are tiny organisms that can only be seen under a microscope. Our own bodies are composed of millions of cells, but bacteria are called unicellular, that is, they have only one cell. Bacteria are usually much smaller than the cells in our bodies.
Also, the bacteria can multiply anyway. It can multiply in the air, it can multiply on the surface. It doesn't really need a host to multiply. So also, bacteria are different. Though they are under bacteria, but they are different. And because they are different, then they attack different body parts. Some bacteria you cannot find in your brain, you will find them in your intestines. And the ones that you find in your intestine, you can't find them in your brain.
Bacterial infections are treated with various antibiotics. An example of an antibiotic is Bactrim or cotrimoxazole, which is used to treat and prevent some bacterial infections. Viruses are even smaller than bacteria. They are so small and simple that scientists are not sure whether or not they can be said to be alive. Viruses have to infect the cells of other creatures before they can reproduce themselves. Other examples of viruses include colds and flu, herpes, and HIV.
What is so unique about the virus is that it cannot multiply without a host. It needs a host to multiply. So it needs people like us in order to make its own babies or to make its own copies. So what it does is, in a case of HIV, it attacks our CD4 cells, one of the biggest important cells in our bodies to fight infections. It attacks them and they multiply in them. Remember, I said a virus needs a host, and it's very small. It can go anywhere in your body. It can go into your blood. It can go into any body organ. That is what is so unique about viruses.
Fungi are like little plants that can infect our bodies. They are often parasitic, that is, they feed off other creatures. Again, not all fungi are harmful. Mushrooms and yeast are also examples of fungi. Parasitic fungi that infect our bodies include athlete's foot, dandruff, thrush, and PCP pneumonia. Ringworm is actually also a fungus, even though it's called a worm.
A fungus also can multiply in the surface. Sometimes you can see it with a mold, but you can't see the mold when it's inside your body, but it will cause an infection. For example, I'll make of some of the ringworms that some people do get.
The last main category of germs or pathogens are called protozoa. Examples of protozoan infection include malaria, toxoplasmosis, and cryptosporidosis. Understanding what kind of germ causes an illness can help you understand treatment better. For example, a cyclovir is used to treat both shingles and herpes because both are viruses that attack the nerve endings. Our bodies can also be infected by larger parasites like tapeworms or bilharzia. Bilharzia worms infect us by actually digging their way into our bodies through our skins. Worms can infect and even block blood vessels in our intestines. They eat our food inside us and use up the energy we should be getting. Up to 90% of children in some informal settlements have worms. Fortunately, treating worms is cheap and easy. And there is some evidence that treating worms alone can reduce the amount of HIV in our bodies.
Different kinds of germs. Bacteria, for example, TB or syphilis. Viruses, for example, HIV, herpes, colds and flu. Fungus, for example, thrush, PCP and ringworm. Protozoa, for example, toxo, crypto and malaria. Parasites, for example, bilharzia and worms. Pathogens, for example, germs in our bodies that cause disease.
Now that we've got an overview of the different kinds of infections, let's look more closely at how HIV works. Every cell in our bodies has a nucleus. The nucleus of the cell contains all the information needed to make a copy of this cell. In this way, cells become heart cells, lung cells, skin cells, etc. To make copies of the cell, a special chemical called DNA is needed. DNA stands for deoxyribonucleic acid, but you can just call it DNA. Think of DNA as the instruction code that tells our bodies how to work. HIV enters the body through unprotected sex, mother-to-child transmission, and in breast milk or injection drug users, needle stick injuries, or HIV-infected blood products. Once in the bloodstream, HIV attaches itself to the surface of the CD4 cell using special proteins called GP120 proteins. The nucleus of the HIV cell that contains the RNA then enters the CD4 cell. HIV changes its language, instructing it how to live from RNA to DNA. To change itself from viral RNA to viral DNA, HIV uses a special protein or enzyme called reverse transcriptase. HIV's DNA integrates into the DNA of the CD4 cell. HIV DNA in the CD4 cell makes new HIV RNA that is released into the cell. New HIV RNA makes new HIV proteins. The new HIV viral RNA are assembled into new HIV viruses. The new viral particles make their way to the surface of the CD4 cell. When they reach the surface of the cell, they use the protease enzyme to bud out of the cell and are released. The new HIV are assembled and bud from the CD4 cell. The HIV-infected CD4 cell becomes a factory for new HIV. Eventually, the CD4 cell does not do its job well and dies.
HIV life cycle. HIV lives in the human body only. HIV attaches to cells using the CD4 cell. HIV attacks the CD4 cells and produces more HIV. DNA contains all the information of the human cell. RNA contains all the information about HIV. HIV converts its RNA into DNA. HIV inserts its DNA into the DNA of the CD4 cell.
This is the end of part A of the introduction to HIV. In part B, we'll go on to find out how our bodies work, and we'll be looking at the different stages of HIV illness.